Welcome back to the channel. In this video, we're going to be looking at the concluding part of aerobic respiration from the point of the biochemical processes involved. We will start off with a recap of aerobic respiration and then of the role of the Krebs cycle and what happens within that cycle. We'll then move on to oxidative phosphorylation, and then we'll talk about the roles of NAD and FAD. And then we'll move on to, very briefly, chemiosmosis, which we'll deal with in greater depth in a future video on its own. And then finally, we'll talk about what proton motive force is and how it generates ATP. So let's crack on with it. Okay, so we're starting with page 6 of the worksheet on the biochemistry of aerobic respiration. So where does the Krebs cycle take place in a cell? It takes place in the matrix of the mitochondria. Within cells. So it's within the matrix of the mitochondria. As you can see there, that's a summary, a diagram showing a summary of the Krebs cycle, which is also known as the citric acid cycle or the tricarboxylic acid cycle. It's slightly more detailed than you need to know. Okay, so what is all the main purposes of the Krebs cycle? So number one, to complete the breakdown of glucose. And number two, to generate reduced NAD, reduced FAD, and some ATP. Okay, so that is the point of the Krebs cycle. Okay, so the next bit. Why does the Krebs cycle occur twice for each glucose molecule in aerobic respiration? So let's have a look back here. As you can see, the molecule that is put in is acetyl coenzyme A, which is a molecule we end up with at the end of the link reaction. And if you remember going back to the previous video, the link reaction has to happen twice for each molecule of glucose because the glycolysis leads to the production of 2 molecules of pyruvate. Each of those 2 molecules is then fed into the link reaction, and so each of those will result in 1 molecule of acetyl coenzyme A. So there needs to be 2 occurrences of the Krebs cycle for each molecule of glucose. So glucose ultimately results in the production of 2 molecules of acetyl coenzyme A. And acetyl coenzyme A is what we're feeding into the Krebs cycle. So what is the job of the 4-carbon molecule oxaloacetate? So if you look back at the diagram, you'll see that acetyl coenzyme A combines with oxaloacetate at the start of the process. So oxaloacetate is responsible for combining with acetyl coenzyme A at the start of the Krebs cycle. So combines with acetyl coenzyme A at start of Krebs cycle. Okay, so what does dehydrogenation mean and what enzymes are involved in the process? So it means the removal of hydrogen from a molecule. And the enzyme, unsurprisingly, is a dehydrogenase. So the enzyme responsible is a dehydrogenase. Okay, so decarboxylation, quite a similar sort of answer for that one. What does decarboxylation mean and what enzymes? So number one, it's the removal of carbon from a molecule. And the enzyme involved is a decarboxylase. So when you remove a carbon atom, the carbon atoms in these biological molecules are very often also combined or bonded to hydrogen atoms. So when you remove the carbon atoms, you also have to remove hydrogen atoms. So generally speaking, at any of the steps in the Krebs cycle where the number of carbons in the molecules drops, you're going to need decarboxylases and dehydrogenases in order to facilitate those changes. So what are the 5 main products of the Krebs cycle? So let's think about this. Let's start off with the most obvious product. In order for any cycle to keep going, in order for it to actually genuinely be a cycle, you must be able to regenerate what you started off with. So you need oxaloacetate. So it regenerates the oxaloacetate, which can then recombine with more acetyl coenzyme A in the future. You also need reduced, or you also produce reduced NAD, reduced FAD, and we'll talk about what those stand for later. You also produce a certain amount of ATP and ADP, depending on which steps. Some steps produce ADP because they require the removal of a phosphate group from ATP. And in some cases, ATP is generated from ADP. It all depends on the situation. And there are other substances which are also released. In fact, actually, let's couple ATP and ADP as one. And then we can then have carbon dioxide as another. Okay. So those are some of the main products of the Krebs cycle. So once the Krebs cycle is completed, the last step is a process called oxidative phosphorylation. And this is the final process of, or the final main biological process or biochemical process in aerobic respiration. This process takes place on the inner mitochondrial membrane of the inner mitochondrial membrane, and sometimes that's called cristae. So the sort of folds within the inner mitochondrial membrane. So the inner mitochondrial membrane, or cristae. Okay, and you can see that in the diagram, the folded inner membrane. How is the inner membrane of the mitochondria or cristae adapted to its function in oxidative phosphorylation? So it's folded to increase the surface area. And has embedded proteins. We'll talk about what those embedded proteins do later on. So describe what oxidative phosphorylation is and what the roles of reduced NAD and reduced FAD are in facilitating it. So oxidative phosphorylation, as the name suggests, it's a process that brings about the phosphorylation of ADP to ATP by coupling it with a series of redox reactions. So a series of redox reactions which generate ATP from ADP. So by phosphorylation. So what do I mean by redox reactions? So in a reaction where you end up with a molecule being reduced, there will also usually be another molecule which is being oxidized in order to bring about the reduction of the other molecule. And as such, we call those redox reactions because they're paired reduction and oxidation together. So what's the job of NAD and FAD? So reduced NAD stroke FAD act as what we would describe as proton, hydrogen ion, and electron transporters. They are proteins. Well, actually, they're let's not say that they aren't actually proteins. They're a different class of molecules. Okay. So chemiosmosis. So as I said earlier, we're going to deal with chemiosmosis in a bit more detail in its own video because it warrants it. It's a bit more complicated than what I would like to do here. But I'll give a very brief description of what happens in oxidative phosphorylation and chemiosmosis and how they're linked together. So the net result of oxidative phosphorylation is the accumulation of hydrogen ions, protons, in the intermembrane space in mitochondria using ATP from the process to bring this about. So the ATP is used to basically force the protons into the intermembrane space. The intermembrane space is the space in between. So if we go back to this diagram, the intermembrane space is the space in between the inner and the outer membrane of the mitochondria. Okay, so back to our summary of oxidative phosphorylation and how it's linked to chemiosmosis. So next, chemiosmosis generates ATP by coupling the movement of hydrogen ions, protons, across a membrane through stalked particles, proteins, associated with an enzyme called ATP synthase. So that's a bit complicated. What's happening is that you have those hydrogen ions that have been allowed to accumulate in the intermembrane space, and you have particles, stalked particles, which are channel proteins within the membrane, which then allow the hydrogen ions or the protons to flow down their concentration gradient. Now that you've created a concentration gradient between the intermembrane space and the matrix of the mitochondria. So what ends up happening is, because you have that concentration gradient, the protons or hydrogen ions then flow through those stalked particles. As they flow through, because the stalked particles are associated with this enzyme, ATP synthase, the movement of the protons, what we call proton motive force, causes the or brings about the phosphorylation of ADP to ATP. So let's put that down as the last point. The flow of the protons supplies the energy required to phosphorylate ADP to ATP. So that is, in a nutshell, how this works, but there'll be a bit more detail in a future video. So you have an opportunity now to pause this so that you have a chance to write this down onto your copy of the worksheet. And as usual, there is a QR code at the bottom there, which takes you to another interesting video on YouTube about this. Okay, so let's go on to the next bit. What is proton motive force? This is the phenomenon which arises due to the movement of hydrogen ions, protons, supplies energy to make ATP. Okay, so why does the intermembrane space have a low pH? So why is it that in the mitochondria, the intermembrane space would have a low pH? Let's think about what pH is. So pH is a measure of the concentration of protons in essence, but it's a negative figure, so it's actually equal to minus log of hydrogen ion concentration. Okay, and that's because the hydrogen ion concentration is an enormous number. And so in order to deal with what would otherwise be an exponential curve, and if you do chemistry, this will make sense to you. It would give you an exponential curve. By plotting log instead, you end up with a straight line. So you can take what would be an exponential growth in numbers and turn that into a linear growth in numbers by using log rather than the raw concentration of hydrogen ions. And because it's a negative figure, you end up with a situation where it's the inverse in essence. So the lower concentrations end up with higher figures, and the higher concentrations end up with lower figures. That's why things that are more acidic and that end up with a higher concentration of hydrogen ions actually have a lower pH. So something like HCl could have a pH of 1, whereas more alkaline solutions, which have a lower concentration of protons, tend to have a higher pH. Okay, so why is it that the intermembrane space has a low pH? Well, the reason why is because we've accumulated hydrogen ions within the intermembrane space. And as I said to you before, the pH is an inverse log of the concentration of hydrogen ions. And as a result, you're going to end up with a figure for the pH within that area, the intermembrane space, being closer to the acidic end of the pH scale. So it's due to accumulation of hydrogen ions, protons, in the space, so in the intermembrane space. Okay, now I would like you, using a source like the internet or a textbook or anything along those lines, to complete that table, the steps and the molecules of ATP generated. I'm hoping that you'll notice something when you go through trying to find sources for this information. And in the next video on chemiosmosis, I'll mention what you would have ended up finding. So what stage is the most important for generating ATP? No matter what source you come up with, your answer will be oxidative phosphorylation and chemiosmosis. So the two coupled together. Okay, so though glucose is the main fuel or substrate used in respiration, other substances can also be used. And so what we're getting at here is that glucose isn't the only thing that can be used in respiration, even though you've been taught in the past that that is the case. But you could use lipids, for instance. You could use other complex carbohydrates. You could use even proteins, so it all depends on what is readily available. Okay, so here on the last page is a summary of the entire process. So at the beginning, you start off with glycolysis. And as you can see in glycolysis, you split glucose into two molecules of pyruvate. That's the point of that, basically. And then so as you can see, glucose starts off with 6 carbons, and pyruvate, you have 3 carbons in each. And then in the link reaction, which is here, you end up with the pyruvate generated from glycolysis being converted into 2 molecules of acetyl coenzyme A, which has 2 carbons. So 3 carbons there and then 2 carbons here with a loss of carbon dioxide. Okay, so the link reaction is there. Okay, now next was the Krebs cycle, or the tricarboxylic acid cycle, or the citric acid cycle, whichever name you prefer. It's probably easier to stick with the Krebs cycle. The point of the Krebs cycle is to complete the breakdown of glucose. So those remaining 2 carbons in acetyl coenzyme A, in each of the molecules of acetyl coenzyme A, are split off, and the whole molecule is now broken down. And as a result, you generate even more carbon dioxide. Okay, so that's the Krebs cycle. And then finally, you have oxidative phosphorylation, which, as I said when we looked at that in the video, is the coupling of redox reactions to the generation of ATP. So you make ATP by adding phosphate groups to ADP, and the source of the energy is a series of redox reactions, as well as the movement of protons across the inner mitochondrial membrane. So during oxidative phosphorylation, why does the transfer of electrons from carrier to carrier generate ATP? This is because the carriers exist at different energy levels, and as electrons move from a higher energy level to a lower energy level, energy is released to make ATP. Okay, so that's a sort of summary of it. And in reality, that ATP is not really what we're trying to get at. What we really want is to use that ATP in essence to bring about the accumulation of protons in the intermembrane space. And because the protons are now accumulated in the intermembrane space by active transport in essence, once you have channels that allow for those protons to then pass through the inner mitochondrial membrane, they will flow through a bit like a dam. So in a dam, you have turbines and you allow water to flow through or flow down due to gravity through those turbines, turning the turbines and generating, for instance, hydroelectricity. In this case, the flow of the protons is a bit like the flow of water to a dam. As the protons flow through those stalked particles in the inner mitochondrial membrane, because the stalked particles have ATP synthase on them, an enzyme which brings about the generation of ATP or phosphorylation of ADP to ATP, that movement generates ATP. So let's talk about what the roles of NAD and FAD are. So NAD stands for nicotinamide. Terrible spelling today. So nicotinamide adenine dinucleotide. Do you need to know this name? No. But I'm mentioning it just in case you come across it in an exam or something, and just that way you don't feel uncomfortable or completely flummoxed by it. So what is NAD's job? I mentioned that earlier. NAD acts as a transport molecule for protons and electrons. I will talk more about that using a diagram that I've attached at the bottom. So what are dehydrogenase enzymes? The name tells you really. These facilitate or speed up the release of hydrogen from molecules. In this case, reduced NAD or reduced FAD. So how does this work? Let's have a look at this diagram. So in this diagram, we're using an analogy of a taxi cab. So what happens is when NAD is in its oxidized state, so before it's reduced, it doesn't have any protons or electrons attached. But you can attach protons and electrons to NAD, and by so doing, it's now reduced in the middle picture. And once it gets to its destination in the membrane, or in the mitochondria, it can then offload its electrons and protons, ready to go off and pick up some more electrons and protons. And now those electrons and protons can then be used in oxidative phosphorylation and chemiosmosis. So the whole point of NAD is to pick up electrons and protons to enable oxidative phosphorylation and chemiosmosis to occur. So FAD, we talked about NAD, now let's talk about FAD. And FAD stands for flavoadenine dinucleotide. Okay, and that's how that works. So what are those molecules? Earlier in the video, I made the mistake of describing them as proteins. They aren't proteins. They're actually derivatives of nucleic acids. So in essence, nucleic acids can be changed slightly in order to make them into NAD and FAD. And so that is it for this video. As I said, in a future video, I will deal with chemiosmosis in a bit more detail because it warrants its own video separately. So let's have a quick look at what we've done so far. So in this concluding part, we talked about the Krebs cycle, which completes the breakdown of glucose by taking an acetyl coenzyme A from the link reaction and splitting off the remaining carbon atoms. So now all the bonds have been broken up from the glucose we started off with. That means that you're obviously going to be generating some carbon dioxide, and because the carbon that's been taken off would have been attached to hydrogens, you're also releasing some hydrogen ions or protons. As it so happens, those hydrogen ions and the electrons that are associated with them will then be transported by NAD and FAD, which is why you also generate reduced NAD and reduced FAD in the process. And you also regenerate the oxaloacetate, which starts off the process by combining with acetyl coenzyme A. So that's the Krebs cycle. The next step is oxidative phosphorylation, and we tend to couple that with something called chemiosmosis, which we'll talk about in the future. So in oxidative phosphorylation, you've got a series of redox reactions, which basically cause the generation of ATP from ADP due to energy being released as electrons are moved between electron carriers. That ATP that is generated is then used to actively pump hydrogen ions, protons, into the intermembrane space. That's the space in between the outer membrane of the mitochondria and the inner membrane, the cristae of the mitochondria. As such, that buildup of hydrogen ions leads to a drop in the pH in the intermembrane space. That also generates a differential in terms of the proton concentration on either side of the membrane. And so because there's now a concentration gradient generated across the membrane, the openings, the stalked particles or proteins in those membranes, will then allow the hydrogens to flow across the membrane by osmosis, chemiosmosis. And because they're flowing through the membrane, that releases energy, which, with the aid of ATP synthase, brings about the phosphorylation of ADP to ATP. And that's how we generate ATP in this process. It all sounds a bit complicated, but what you need to do with anything like this is just keep coming back to the video and refilling the worksheets, and eventually it will stick. There isn't really a sort of magic way to learn this. I'm not some special genius or something. It's just about sitting down and getting on with revising this over and over again until it sticks. Okay, so as usual, if you found that useful, please do click on the like button below, and if you haven't done so already, subscribe to get notified when I put up more videos in the future. So I wish you the best of luck, and hopefully I'll see you again in a future video.